So now we're going to move from determining the effects of mutations to repairing them. This will go into our DNA repair mechanisms, and we're going to refresh our minds on what we just did in the previous challenge question with the types of mutations, but now we're going to use our different repair mechanisms to repair them, and use this as a bridging point to talk about DNA repair mechanisms. So in the previous slide, or the previous video, we talked about a transversion at nucleotide 10, a transition at nucleotide 12, and a transition at nucleotide 7. These are all examples of point mutations where a single nucleotide has been altered or changed in some way, as opposed to chromosomal mutations that we talked about in Unit 2, such as translocations, deletions, inversions and things. Point mutations affect single nucleotides. So the first one says that transversion in nucleotide 10, let's fix it by mismatch repair. And mismatch repair is the first DNA repair mechanism that we talked about in the second lecture of this week. Now, if I'm told that I need to fix this by mismatch repair, I automatically know a few things about this mutation. I know that it's a spontaneous mutation rather than induced. It was caused by a cellular error as opposed to something from the environment. The reason why I know that is that the mismatch repair pathway is reserved exclusively for replication errors, errors made by DNA polymerase during DNA replication. Those errors are cellular errors, they're spontaneous mutations, and so mismatch repair is only responsible for those replication errors. Mismatch repair cannot fix or repair induced mutations that are caused by the environment. An important point there. So mismatch repair. So here's our double-stranded DNA, and it's anti-parallel, and it's base paired, and it's nice and smooth. It has that beautiful smooth configuration, that very repeating structure. And when we have a mismatch error, that is to say when DNA polymerase has made a mistake and it puts the wrong nucleotide across from its partner, as in this case it did for this transversion, that mismatch, that, how should I say it, improper base pairing distorts the smoothness of the DNA topology. And there are proteins, enzymes, whose job it is to constantly move up and down our chromosomes, feeling for the smoothness of that DNA topology. And anytime one of those enzymes comes upon a bump or a distortion in that smoothness, it stops and it marks that region as needing repair. So that's what happened in the first steps of mismatch repair. In fact, I should say that's what happens in the first steps of all the repair mechanisms. And so mismatch repair is triggered by this distortion, by this irregularity in the smoothness of DNA. Now, in order to tell the mismatch story, we have to remind ourselves of a few things that are unique to this pathway. First, at least in bacterial cells, old DNA, DNA that's already been checked and repaired, is methylated quite frequently. Methyl groups or CH3 groups are added to the DNA itself. So if this is a mismatch error, if this is a DNA replication error, that error was made on the new, just synthesized strand of DNA. What that means to the cell is that the methylated strand of DNA, the bottom strand here in this diagram, is the older, already fixed, repaired, and checked strand of DNA. It is the trustworthy strand. And if the bottom strand here is the trustworthy strand, it's already been quality controlled, then that means the reason for the bump and the reason for the distortion lies in the unmethylated strand. It lies in the strand of DNA that is missing the methyl groups. The unmethylated strand is the new strand of DNA. And if this was a replication error, the error was made while making the new strand of DNA. So what the cell does in order to repair DNA by the mismatch repair mechanism is it nicks the DNA backbone on the unmethylated strand at the nearest methyl group downstream of the mutation, the nearest methyl group at the 3' end of the mutation, which would be this methyl group right here. And then once that DNA is nicked, the DNA is then slowly chewed back all the way to the distortion so that the distortion is completely removed. And then a unique DNA polymerase called the repair polymerase lands on this open end, this single-stranded end of the repaired DNA. It lands there, and it makes new DNA filling in that gap. And what you see is that once that's complete, all we are left with is what's referred to as a nick in the backbone of the DNA. There's a nucleotide there. We're just missing the phosphodiester bond that links the nucleotide right here to the next nucleotide right here. So to fix that nick, or to repair that break, to weld that DNA together, we need a unique enzyme called DNA ligase. And DNA ligase is also common among all of the repair mechanisms. DNA ligase lands at this nick, makes that phosphodiester bond, sealing the nick shut, and now the mutation has been completely repaired. So that's the mismatch repair mechanism. We've got a methylated old strand that has already been deemed trustworthy. We have a very newly synthesized strand because mismatch repair is only used for DNA replication errors. And if there is a distortion after DNA replication, it is because of a mistake on the new strand, the unmethylated strand. So the new strand is nicked immediately across from the nearest methyl group, chewed away all the way to the distortion. DNA repair enzyme comes down, DNA repair polymerase comes down and puts new DNA in that gap, and then DNA ligase seals it all shut. The last step of this process, while it's not technically part of mismatch repair itself, is that once the new strand has been repaired and all those replication errors have been removed and replaced, it's time to deem the new strand as trustworthy. And so methyl groups are added to the new strand, marking it now as old and trustworthy, so that in the next round of replication, this repaired strand can be used as the template. So that's mismatch repair. Now the next two pathways of repair, which are base excision repair and nucleotide excision repair, are for induced mutations. These are for the environmental mutations that we pick up just from our outside environment. So mismatch repair is exclusively for spontaneous mutations, and base excision and nucleotide excision are for induced environmental mutations. But they all start the same way. Remember, I told you before, with mismatch repair, every single one of these repair stories starts with one of these scanning enzymes that detects the mutation because of a bump or a glitch in the smoothness of the DNA topology. So in base excision repair, that bump is sensed, and DNA cutting enzymes are recruited, and they cut just in front of and just after the bump. Now, to be fair, because there's no methylation in this process, it is possible for these DNA cutting enzymes to get it wrong and to cut the non-mutated strand and actually repair the DNA in the wrong way. That's actually how mutations become locked in our genome, is in this way. Since there's no way to tell what the good strand is and what the bad strand is with induced mutations, essentially cells are rolling the dice and picking one strand randomly and repairing it. But in either case, the DNA backbone is cut immediately before and immediately after the single base pair that's causing the distortion. That base is removed. DNA repair polymerase lands on the open end of the gap. A single new base is put down across from the original, using this original strand as template. We've got a nick in the backbone. Something needs to come in here, land, and weld it shut. That is always going to be DNA ligase, and base excision repair is complete. That's all there is to it. For nucleotide excision repair tends to be reserved for more highly mutated areas, for areas where there are multiple bumps or distortions. So still induced errors, but here the DNA is not really distorted in one region, but distorted in multiple regions. So what happens in nucleotide excision repair is that those distortions are first sensed by a DNA scanning enzyme. Let me just fix this here. And then once sensed, this DNA is actually pulled apart. It is made to be single-stranded. The entire region of DNA that seems to be showing these distortions is separated. The two strands are separated. So we almost get like this bubble of single-stranded DNA regions. And then DNA cutting enzymes come in, and they cut one of these strands at the end of the bubble. So this entire region is liberated. At that point, as you can probably guess, DNA repair polymerase comes in, lands on this end, and uses this strand as template, finishing until all that remains is a nick. And then DNA ligase comes and seals the nick. Once the DNA has been completely restored, of course, it can assume its normal shape. It doesn't remain bowed in the way that I have it here. And the entire offending region has been removed. So you can see some commonalities between all three repair mechanisms. All three require these scanning enzymes to sense the presence of mutations by distortions in the shape or smoothness of DNA. Then some differences. Mismatch repair is the most involved mechanism we go over with the methyl groups and the differentiation between old and new strands. Mismatch repair exclusively for replication errors, spontaneous errors. Base excision repair and nucleotide excision repair, very, very common to each other. We have this nicking of one strand before and after the mutated region. The nicked region is removed. DNA repair polymerase comes down and adds new DNA, which is true for all three mechanisms. DNA ligase seals the nick, which is true for all three mechanisms. So students often get confused by base excision repair and nucleotide excision repair because they are so similar. So here's a nice simple way to tell them apart. This is the way that I remember them. So again, the B stands for base and the N stands for nucleotide. And I tell them apart by remembering that the small word is responsible for the smaller repair. You just remove the single base that's offending, the single base that's mutated. And the bigger word is for the bigger repair. This is when we remove multiple bases at once and put down a whole long strand of DNA. So that's how I keep them apart. Hopefully that works for you. And that's our quick tutorial on the different DNA repair mechanisms.